So let's get started on this last session. Our first speaker is Professor Shinya Fushinobu from the Department of Biotechnology at the University of Tokyo, where he has been on the faculty since the late 1990s, and he served as full professor since 2012. He earned his PhD at the University of Tokyo and trained as a structural biologist, including time as a visiting scientist at Iowa State University. Professor Fushinobu is internationally recognized for his structural and mechanistic studies of carbohydrate-active enzymes, particularly glycoside hydrolases. His work has shed light on how gut bacteria digest complex glycans, how enzymes evolve new specificities, and how unique sugar chemistries, such as those found in plant walls and microbial cell surfaces, are recognized and processed. And today, he's going to, I still have a few sentences, he's going to present on structural analysis of enzymes degrading mycobacterial lipoarabinomannan and arabinogalactan, describing recent crystal structures that reveal how soil bacteria break down unusual araban linkages found in cell walls of Mycobacterium tuberculosis and related pathogens. This research not only deepens our understanding of enzyme diversity, but also provides insight into the biology of one of the world's most infectious agents. So we're looking forward to your talk.
Thank you very much, Professor Mascazas. And also, I'd like to thank the people from University of Minnesota and also for Amano Enzyme for giving us a chance to talk about our recent research. And my name is Shinya Fushinobu, born in Hiroshima in Japan. And this is my Twitter X account with 4,000 followers. And I frequently tweet. And sometimes I tweet some kind of a politically incorrect tweets. So, but I was lucky to come here to get across the border. Anyway, let's start my talk. I will start my talk from the cell wall structure of mycobacteria. You know, mycobacterium is known for the tuberculosis or leprae. Those are pathogens, and this is a non-pathogenic strain of the mycobacterium. And they are also called acid-fast bacteria. That is because these kind of microbes are very tolerant, has a very unusual feature of the staining due to the cell wall structure. The outside is covered by mycolic acid. This is very hydrophobic, right? And also it has a very complex cell wall structures, especially for the carbohydrates. These molecules called lipoarabinomannan, LAM, and arabinogalactan, AGs, have a very complex carbohydrate structures that can connect the cell wall components like lipids and peptide glycans, also cell membranes. Right. The main carbohydrate component of these molecules are made of the D-arabinofuranose. You know, the L-arabinofuranose is very abundant on the plant cell wall, but this is a mirror molecule, enantiomer of the D-arabinofuranose, right? And they are mainly made of alpha bonds, but some kind of beta bonds, right? So this is a chemical structure of the LAM and AG. You know, there are more than 22 arabinofuranose sugars are connected to form the D-arginine central core structure. And it is also linked to the lipoarabinomannan or peptide glycan or mycolic acids or some other molecules. Anyway, this is the biosynthesis of these molecules have been very done. But also this molecule is very interesting target, challenging target for the organic synthesis. So these our collaborators, Dr. Ishiwata and Ito, accomplished the synthesis of these big molecules, right? But however, the degrading enzymes of these molecules have not been identified, right? So back in the 1970s, the D-arabinonase was discovered from the soil bacterium called Coccobacillus strain M2. And the enzyme was extracellular enzyme that is induced by the arabinogalactan, right? And this was reported by the Osaka University Dental School, Professor Shozo Kotani and his group colleagues in 1971. In this year, I was born. Anyway, the journal name is called Biken Journal. Biken means, B means Biseibuts, microbe, and Ken means Kenkyu research. So this is a truly Japanese result, right? But there was no gene cloning, so the enzyme was a lost enzyme for 50 years. And also, this is a point that this is a soil bacterium, but its name changed 3 times, right? At first, it was M2, just M2, and it changed its name to Pseudomonas. In 1993, it changed its name to Aureobacterium arabinogalactan, right? Very long. And after that, finally, it has this name, confusing name, mycobacterium arabinogalactan. It is very similar to the mycobacterium, right? So this is very confusing. So that's why we call it, we describe it as a MI strain to distinguish it to the mycobacterium, right? Our collaborator, Dr. Kiyotaka Fujita, tried to find out the identity of those lost enzymes by purifying the extracellular enzymes by chromatographic columns by chasing the activity that degrades arabinogalactan from Mycobacterium smegmatis. And after the purification, the purification was very good, but he could not determine the N-terminal sequence. He found the candidate from the draft genome sequences to find out the ORFs possessing the cell sequence and matching molecular weight and PI values. So they found that DUF 4185, DUF means domain of unknown function. DUF 4185 is a candidate because this gene is also present in the genome of the Mycobacterium smegmatis, because whose endoarabinose activity was reported before, right? So there are 4 genes, the DUF genes in this strain, and they form a gene cluster in a short region, right? 1, 2, 3, 4 enzymes are found, and they are also associated with the glycosylated genes, GHs. And also they are expressed with induction by LAM, right? So after that, this DUF family was named to glucosyhydrolase family 183. So they characterize these enzymes. At first, the cleavage site specificity of the 2 endoenzymes called endo-MA1 and MA2 were studied by using synthetic oligosaccharides to find out the cleavage site for the both enzymes. The sites are very similar, but they found that this heptasaccharide is very resistant against endocleavage, right? So these other 2 enzymes are working in cooperation with these endo-type enzyme. These 2 are actually exo-type enzymes. We named them as ExoMA1, ExoMA2. They belong to different GH families. At first, GH172 enzyme, ExoMA1, is a D-arabinofuranosidase, alpha D-arabinofuranosidase that can cleave the alpha bond of from the non-reducing terminal of the oligosaccharides, right? And the other one belongs to the GH116 family, where the ExoMA2 can cleave the beta bond from the non-reducing terminus of the oligosaccharides, right? So these 3 enzymes, one is endoenzyme, the others are exoenzyme with a different linkage specificity, can collaborate to degrade the core D-arabinon complex D-arabinon structure into the monosaccharides with a cooperated action. Okay, so this is the overview of the degradation pathway of the microbacterium arabinogalactan liticum. And the two enzymes have signal sequences that are, so they are extracellular enzyme that can break down the polymer into the oligosaccharide. And there is an ABC transporter genes right next to these enzymes. So maybe the ABC transporter can incorporate the oligosaccharide. Then the other two enzymes, they don't have a signal sequence, so they are intracellular ones, enzymes. So they can break down into the monosaccharides in the cells of the mycobacterium, right? Right. Our study is about the 3D structure determination of these arabinoid degrading enzymes. I will talk about the structural features of these three enzymes. We determine so many kind of crystal structures, and one for the cryo-EM in complex with some ligands. Right. At first, I will start from the GH183 endo-dearabinase. Endo-MA1 structure. At first, we determined the crystal structure of this enzyme, but we found that there are 4 molecules, tetramer, in the crystallographic asymmetric unit, but the interface analysis shows that they could be present as a dimer in solution. But from the analysis using the size exclusion chromatography, the monomer is 52 kilodalton, but there must be some interaction with a matrix that is made of the dextran and agarose to show the very small molecular size, so it's incorrect. So we use the small angle X-ray scattering to find out the real molecular structure in the solution using the combined system of a size exclusion and Sachs system to find out the very clear result that this is actually forms a dimer in solution. And yes, this is the overall structure of the GH183 endoenzyme. We co-crystallized with the monomer of the linear ligand substrate, but in the crystals we only observed the tetramer in the crystals, 1, 2, 3, 4 sugars only, both in the catalytic domain, and we also found the another domain could be a putative carbohydrate binding module, right? It is non-catalytic, but it may bind the sugars polysaccharides on the surface. And yes, we focus on the catalytic domain to find out the cutting site. We use a mutant enzyme that do not have activity to get this complex structure. There are 4 sugars bound in the catalytic domain. This is the nomenclature of the binding site of the glucose hydrolysis. There is a cleavage point between -1 and +1, and we also found 2 sugars of the non-reducing terminus from the -1, -2, and -1, right? And there are 2 catalytic residues. The one is a nucleophile residue that can attack the nucleophilic to the anomic carbon of the sugar, and the other one works as a catalytic acid in the first reaction step. In the intermediate, this nucleophile forms a covalent intermediate. The next time, this D51 works as a base residue to activate the water molecule to detach the sugar moiety from the enzyme. Right. There are also another acidic residue that could be catalytic residue because all 3 mutants of all 3 residues lost the activity less than 0.01%. But the third one is not actually a catalytic, but it assists the ligand binding behind the arginine residue, the important arginine residue, right? So next, I focus on the overall structure of the catalytic domain of this enzyme, right? There are two domains, and the catalytic domains has a 5-bladed beta-proper structure that is similar to another family of enzyme. It's called GH43, but this enzyme is alpha L-arabinofuranostase with an animal inverting activity. So it's very interesting coincidence that these two proteins have a very similar overall structure with an enantiomer substrate cleavage, right? And also we focus on the putative carbohydrate binding module in the endoenzyme. No, the arabinone tetrasaccharide was bound to the very shallow groove on a non-catalytic beta sandwich domain associated with the catalytic domain. And these are for saccharides, these are bound in a very specific way. And structural comparison of this CBM to other CBMs could show an interesting feature because, this CBM has two clefts. One is a major cleft, but it was empty. The another one, shallow cleft, was occupied with arabinofuranose oligosaccharides. So this CBM is similar to two kinds of CBMs. One is for CBM4 family, the other is CBM61. This one, CBM4, is occupied by the xylo oligosaccharides with a major groove. And in this case, malto oligosaccharides were bound to both of the clefts. So we think this domain is a new kind of carbohydrate module, new CBM. So now we are analyzing the binding ability of this domain using the gold nanoparticle system. Using a glycan-bound glycol nanoparticle can be made by Professor Yasuo Suda and Hiroyuki Shinji in Kagoshima University. They invented this system to find out, to use it for the visual analysis by aggregation of the GNP beads. This enzyme forms a dimer structure. So we use the non-catalytic mutant to analyze the binding ability of this domain. And in this case, DP with polymer glycan and longer glycan chains can bind to this protein, but a short one or L-arabinose could not bind to this domain, right? So this could be could be used for potentially application for easy detection of mycobacteria cell wall, right? This is a phylogenetic tree of the GH183 enzymes, proteins. These endo-MA1 and endo-MA2 were the enzymes we found in this study. And one of them, endo-MA1, was only associated with a CBM domain. But most of the enzymes, GH183 endoenzymes are associated with another, D-arabinone degradation enzymes. They make a gene cluster. But interestingly, they are also found in the mycobacteria. This is an internal enzyme, but they are not associated with other degradation enzymes. So this enzyme could be involved in the cell wall remodeling of themselves. Okay, let's go to the next enzyme. This is a family number 172, exo-MA1. This is an exotype enzyme. And the crystal structure shows that it forms a dodecamer. 12 molecules are associated to form a big molecule, looks like a tetrapod, water dissipating concrete unit. I mean, it looks like this. But anyway, but the monomer has a very interesting structure. This is called a double jelly roll fold. This is very rare in the case of the glucose hydrolysis. So after that, they think that they could have an evolutionary relationship with the viral capsid proteins. Anyway, I don't know what is the origin of this protein, but it actually has activity to cleave of the alpha D arabinofuranose in the X way. And the active site is like here. And this enzyme, the another enzyme in the same family, 172, we previously found the enzyme from Bifidobacterium. It can cleave as alpha D fructosidase activity, right? And this has a different kind of oligomer structure. It's a hexamer structure, but monomer structure has a similarity. The active site is here, and in the active site, this enzyme can cleave the fructofuranose because of the very open active site. But in the case of the D-arabinofuranoses, this residue is asparagine. It can block this point so this steric hindrance can inhibit the cleavage of the fructofuranose, but it's very active on the arabinofuranose, right? Because this enzyme has a big structure, and also the crystallization reproducibility was very bad. So we used the transmission electron microscopy to observe the molecular structure. The traditional negative stain TM can show the very clear structure. So we also use the cryo-EM to determine the molecular structure of them, right, with a very good resolution. So the molecular structure of the second enzyme was very clear, right? So this is the phylogenetic tree of this enzyme. This is the one, XOMA1, we studied, and it is also similar. But it is in the same family. There is a fructofuranose enzyme, but in majority, this family is made of the D-arabinofuranosidases, right? Okay, let's go to the third enzyme called XMA2. It belongs to the GH116 family. The enzyme forms a dimer that is connected by so many magnesium ions, and the monomer looks like this. This is a catalytic alpha-alpha 6 domain associated with a non-catalytic beta sandwich domain. And in the active site, we found a very clear electron density of a beta D-arabinofuranose at the -1 subsite. So this is the molecular structure of the active site. And we could compare the structure with another GH116 enzyme called TXGH116. This is actually beta glucosidase. The overall structure was very similar, but it has a different activity that can cleave the glucopyranose structure, right? So as looking at the active site, only the active catalytic residues were conserved. But these residues are completely different to recognize the furanose or pyranose in this case, right? So maybe these enzymes could have a same evolutionary origin, but to become a different, to obtain a different substrate specificity in the molecular evolution. So this XOMA2 enzyme can cleave the terminal beta-2 bond, as I said before. So next time we could determine the disaccharide complex structure filled with a -1, both -1 and a +1 residues using a mutant enzyme to find out the clear molecular recognition at the +1 subsite, right? Also, we found an interesting feature. We found an interesting feature in this enzyme because in the active site, there is in addition to the active site, there is a second site that is located far from the active site. We thought that this could be the second site to bind this branched substrates. I mean, this is dedicated for the branched substrates. So we use a synthetic heptamer to get the complex structure. This is a heptamer structure. The reducing end was blocked by the acetonide substituent, and there are two non-reducing ends. This is a branched one, SC1, SC2, and we could determine the structure occupied in this way. The -1 subsite is occupied the natural residue, and the branch is bound in this way to deviate from the normal binding sites. So this is another view, right? This is the catalytic site -1, and this is the reducing end, right? So the branched residue is located here. It's a bit far from the second site of the enzyme. So maybe this enzyme could have a ability to bind this branch substrate to recognize the branch residue with a second site or this another pocket to bind the branched substrate. So because this unit is very tolerant to the endo type enzymes. So these enzymes can work as in collaboration with these enzymes, three kind of enzymes. Okay, this is the conclusion. There are 4 novel enzymes are discovered by the soil bacterium, which produces lambda-degrading enzymes, right? And there are 4 enzymes and 3 kind of enzymes cooperatively degrade the D-arabinol moiety of the lipoarabinomannan and arabinogalactan in acid-fast bacteria. And we determined the crystal structure of 3 of them. And the first one, endo-MA1 and MA2, were endo-type arabinonases that belong to the new family, G183. Another group in Europe and the US independently discovered this enzyme. The paper was published back-to-back. And the endo-enzyme have a putative carbohydrate binding module domain. And the X1 enzyme could have a novel enzyme with a reduced alpha-arabinonephronase activity. Third one is a novel kind of exo-beta enzyme, right? And this is acknowledged figure. I thank the people in our laboratory, Chiho Nakashima and German Lee and Kashima and Fukushima and Akai for their efforts. Thank you very much.
Thank you very much. Do we have time for some questions? I'll ask a question about the cooperativity of the. So if you remove one of those enzymes, does it completely drop the ability to degrade the polysaccharide or the oligosaccharide, or is it just impaired?
Impaired? Dropped? What? I'm sorry.
So there are 4 enzymes that cooperate in breaking down the cell surface oligosaccharide.
Yes.
So if one is removed, is it completely stop or just slower?
All right, good question. Only the endo enzyme can drop the Yes, only the endo enzyme can leave the oligosaccharides, right? Only the exoenzyme could be also not good enough to degrade because they cannot degrade the longer polysaccharide.
Okay. Thank you. Any other questions? If not, please join me in thanking the speaker one more time.
Thank you very much.